Hi, it's Mr. Anderson. Welcome to Biology Essentials video 28. This is on the cell cycle, mitosis, and meiosis. In other words, how we go from one cell to all the cells, the trillions of cells inside our body. Meiosis is important because that's how we make sex cells. Now, let me digress a little bit. This is a whiptail lizard from the desert Southwest. And what's interesting about this is that it's a female lizard, and when it wants to make more lizards, it will simply use mitosis to make an exact copy of the cell inside its body, called parthenogenesis, or virgin birth, and it will make a brand new number of baby lizards, and they're all females, so they don't have males. It's rare to not have males, it's rare to not have meiosis, and the reason why is that all the whiptail lizards are genetically the same, but it works. If you live in a fairly stable environment, it tends to work. Now we're not going to go into the specifics of mitosis and meiosis. You can look, I've got videos on each of those that talks about the different phases. What I want to talk about this is, in general, what do mitosis and meiosis do, and how does the cell cycle work, and how is it controlled?
What the hell is wrong with you?
How come has me your asshole? Human cell,
this is not the asshole. You're right there. No cooperation.
And what that means is it has two complete sets of chromosomes. So, for example, in humans, 2n equals 46.
Oh,
that means we have 46 pairs of chromosomes.
Guys,
and so the goal of the cell cycle in mitosis is to make a copy of that cell. In other words, to make a diploid cell.
This
diploid cell can enter into the cell cycle again,
not good, guys.
Make more cells and more cells.
Oh my
so
goodness,
the way that we make new cells in the body or replace cells that are damaged is mitosis. In meiosis, we're going to take a normal diploid cell. We're actually going to make a haploid cell. We're going to make sperm and egg. So in humans, N now equals 23. Now, if we were to just stop there, we wouldn't have diploid cells anymore. But fertilization, where egg meets sperm, is going to combine those two cells to make a diploid cell. And now that diploid cell can enter into the cell cycle again. And so we've got kind of two loops. We've got the mitosis loop and the meiosis loop. Mitosis is used to make all of the cells in our body. Meiosis just makes gametes or sex cells. I also in this video want to talk about how we control the creation of
college right there.
There is it. And how we use cycling, an example of that would be the mitosis promoting factor to control the cell cycle and where it is.
This is our number one. What the hell?
Now, if you're talking about cell cycle, the best place to watch is with videos of actual cells
around the world.
And so this is a cell undergoing division.
Now, 4 million main states.
So we start with one cell and make an exact copy of itself.
Actually,
now when you're watching it, let me go back a second. On these first two videos, what you don't see is everything that actually happens before the cell makes a copy of itself. In other words, before the cell is actually able to divide, it has to duplicate all of the DNA and all the machinery of the cell. This last one, we're looking actually inside the cell itself. And so the cell division actually has two parts to it. Part one is going to be the division of the nucleus. And we call that, in general, mitosis. And then after we divided the nucleus, you'll see the chromosomes actually separate here. Then we actually have the division of the rest of it, and that's called cytokinesis. Cytokinesis is the break apart of all the other parts of the cell. So the machinery of the cell, the mitochondria, cytoplasm, things like that. So let me see if I can start that. So first of all, there we go. First, we have the division of the nucleus. You can see the chromosomes pulling apart, and then we have cytokinesis. And so all mitosis is one cell forming two cells, and those two cells are identical to that first cell. And that's how we go from that first fertilized egg inside us to the trillions of cells that we have inside the body. So when you're replacing cells in your body, you're doing it through
do not go near.
So let's look at the cell cycle.
What's
going on up there? Is you'll have a cell enter into this. So a cell is going to look like this. It's going to enter it into the cell cycle as one cell.
Never dying or decaying.
And then it's going to eventually exit out as 2 cells. Each of those cells, remember, can go back into the cell cycle. And so this is how we make all the cells in our body. Now, I've heard that a lot of the dust in the room actually is dead skin cells. And this is what skin looks like.
Really?
So skin is going to be created new cells, and then they're going to migrate up to the surface, and then we're eventually going to lose skin cells.
I know that what I saw.
But we keep replacing those cells, and to do that we use the cell cycle. So let's look at the parts of the
house like that.
If we start first with that cell entering in, the first thing that it'll do is it'll actually enter into the G1 phase. G1 phase, the cell is actually going to grow. It's going to get bigger and bigger and bigger and bigger. We then enter into the S phase. During the S phase, we're going to actually, using DNA replication, we're going to copy all of the DNA inside a cell. It then goes into the G2, or the growth 2 phase, where it continues to grow and gets ready for division of the actual cell. And so G1, S, and G2 are all part of what's called interphase. And if you look at a cell, it's generally in interphase. It's growing, it's copying its DNA, it's growing, or maybe it's just working. It's doing what a normal cell does. And so most of the life of a cell is in interphase. It's in these 3 G1, S, and G2. And the actual copying of the nucleus and copying of the cell, this mitotic phase, is actually going to be really, really small. If it never divides again, it'll actually stall out in something called the G0 phase. And so we've got cells inside our body. You've heard of this, maybe cells of the central nervous system, muscle cells, for example, that never copy themselves during your whole lifetime. They're in what's called the G0 phase, or they're just waiting, and they're not going to make division.
What? I'm not really.
Know when it's time to divide, and when it should go on and when it shouldn't. The best analogy I can think of is it looks kind of like an hourglass. And so there are little proteins inside a cell, and as those proteins accumulate, throughout the life of the cell, eventually you get a critical number of these proteins at the bottom. And once we have enough of those, then it actually tells the cell to advance to the next stage. And those proteins are called cyclins. And so let's look at what cyclins look like. The cycle represented here it's simply as a protein. But if we look here at the G1 phase, the S phase, the G2 phase, and mitosis, there's a set of cyclins, or a set of these different types of proteins. And what they're going to do is they're going to build up. And so cyclin A and B are ones that I'm really familiar with. Cyclin A will actually build up during the S phase, and then it'll drop off right as the cell divides, as it goes into this mitotic phase. And so cyclins will actually accumulate. And so those are like the sands through an hourglass. They're going to get more and more and more and more cyclins as the cell goes through the cell cycle. Now, the other chemical that I want to talk about is something called CDK. CDK is simply a cyclin-dependent kinase. And a kinase is simply going to be a chemical that can speed up actions within a cell. And so CDKs are found in all living organisms. And you can actually take CDKs from a yeast and put them in our cell, and they work just as fine. So they show homology through evolution. And so cyclin-dependent kinases, if you look at their name, are simply dependent on cyclin. I made a little animation of how that works. And so a typical cell in your body is going to have a bunch of CDKs in it, or cyclin-dependent kinases. And so we could say this is like right here. But throughout the life of the cell is going to start building up and accumulating larger amounts of cyclin. So the amount of cyclin is going to get larger and larger and larger. So eventually what happens is the cyclin is going to fit into the cyclin-dependent kinases. Now we have an activated CDK cyclin complex. What does that mean? We have something, a protein that's able to do things. So now you can think of we've like mustered this army, and now the army is ready to do something. And so what does it do? Well, we're right here, so we're just about to enter into the end phase. We're just about to do mitosis. And so what these cyclin-dependent kinases do is they act on the cell itself. So they are a specific type of CDK. It's called the mitosis promoting factor, or MPF. And what that does is when it builds up enough of these cyclin-dependent kinases, they're actually going to work on the cell. So one thing they'll do is they'll actually break apart the nucleus. So we were able to
However
start
Spanish or however
dividing
we-
that cell.
when
Another thing
Brazilian,
that CDKs will do is they'll actually
there's
work on the
London,
microtubules to
British,
build this spindle.
we find
And
United
so
Kingdom
all of
and
these
the
together
United
will
States
work
and the
on
US.
pushing
Wait,
that
US?
cell
There's a
into the
get
mitotic
them
phase,
like
or into
there's
this
a
next
lot.
step,
What's that
step
supposed
of
to
the
mean?
cell cycle.
Never
The neat
done
thing
your
about
vocabulary.
each of these is
What
that
the hell
after
is
they've
that
actually
phrase?
done that,
Get
they'll
out for
actually
me.
gobble themselves
That's
up, they'll
everyone's
disappear, and then the
knowledge,
whole
and
cycle
everyone's
begins over
choice.
again.
You gotcha.
Okay,
Stop right
so if
there.
we kind of talk
Alright.
big
Here's
picture
one
about what
here.
happens in the cell
Okay,
cycle,
what the
a
hell?
typical cell right
Lucas,
here
you're the
is
one
going to be,
here.
let's say this is a typical cell.
No,
A typical cell right
absolutely
here is
not.
actually going to be
No,
two
it's
ends,
like
it's diploid. You have one
no,
chromosome
I
from
win
mom,
the
one
game.
chromosome from
I
dad.
went there.
So in
I'll
this
be
case,
right
it's going to
back
be 2n
before
equals 2.
No.
It's then going to
Absolutely
duplicate
over here.
itself. So
No.
during the S phase,
This
it's going
is
to make
a-
copies
No,
of itself. And so
it
at
is
this point
not
right
one
here,
here.
we'd actually have
I
a
have
4n cell. It's
No,
make copies
I
of it.
ran alone here.
And at this point, we can either take
I'm
the
not
path
gonna
of
get that.
mitosis
Right
or the
there.
path of
No, I'm
meiosis.
not really
And so in the
sir,
path
please.
of
And
mitosis,
please
that'll
call
simply
me now.
split in half,
It's your dangerous.
and now we'll have
This is an
two
emergency.
2n cells. And if you look at these
That's
two
nothing.
cells, two 2n
One.
cells, they're
What is
exactly the
real
same
here?
as that
Means
first cell.
here
So this is
forever.
what's happening
Never
to the
dying or
chromosomes.
decay.
If we look
This
at that
means forever.
4n cell, and
Never
as it
dying
goes
or
into
decay.
mitosis, it'll
Okay,
actually
let's-
line up.
Do you have
It'll
any
split in
right
half,
there?
and then
No,
it'll split
you
in half
wasn't.
again. And so
I'm
what you
just
have
gonna
is actually
forward
N
this.
cells. Those are called haploid cells.
What
And we have
was that?
4 of those
Phrase.
in meiosis.
I know the one.
And so
what was
the
that?
cell cycle will
Phrase.
take a
You
diploid
got
cell,
it,
make
but
2
if
diploid
you
cells in
are
mitosis,
like
or
that,
make 4 haploid
and
cells
Jimmy
in
down the
meiosis.
most.
That got a little messy. So let's look at it in
You're
a little bit
maligning.
more detail.
We don't have
So
any trust me.
mitosis is
We
how
don't
we
have to
replace
learn
cells
how
in
to
bodies.
carry
So cells that are
real
broken,
here.
cells that are
Stop
broken
right
down,
there.
cells that we need to
You're
replace. We do that through mitosis.
me
Or when we want to grow, how do we go from a very small
you see
organism to a very
Ryan?
large organism? It's just
Really?
making more cells.
Isabella.
And so this would be a real,
My
to make it
friends here.
simple, we're going to start with a
Oh my
simple cell.
goodness, look
This
at
is going to be a
Jack's place in
2n,
the
it's
fortune.
diploid, but
Could
2n
be
equals
anything.
2.
But
So this would be that
you're
first
anyone.
parent cell. It'll then duplicate
right
all of
there.
the DNA. So now we
I'm
have this
not
characteristic
right there.
shape.
I'll be
It has
right
2
back.
sides to it,
I'll be
and this
right
side
back
and
before
that
I know
side
it.
are
Hey,
copies.
let's
That's
go
what happened during
happy
the S
world.
phase.
All right, good
They'll
luck.
then meet in the middle. They'll divide in
good
half. So
day.
this would be our mitosis
good day
phase.
here,
And now we have
forever.
2
I'll
diploid
be
cells.
right back before I
And if you
know
look
it.
at those cells, they're exactly the same
You
as
have
that
no
original
knowledge.
cell. And
I
each of
get
those are
knowledge
2n
power like this.
equals
I don't to
2.
get this
So that's
knowledge.
mitosis. What happens
Me?
to these 2n
What is
cells?
a
Well, they can
church?
enter into the cell cycle again.
Church?
It goes over and over
I
and over
thought
again.
that there are so
In
much.
meiosis,
What
what
the hell
happens
is
is a little
wrong
more
here?
detailed.
Like,
Remember,
yes, you don't
meiosis
want me
only
here.
deals
No,
with
we're
making
not
sex
really
cells
young.
in reproduction.
Now,
And so in
no.
this case, to
There's
make it
a me.
to show you
Me?
meiosis, I had to increase
Now,
the number
you
of cells. In this
want me?
case,
No,
4 chromosomes.
stop
2n
that.
equals 4.
All right,
In
stop
other
right
words, we have 2
What is it
chromosomes from
real
mom,
here?
2 chromosomes from dad.
From
So they'll
chicken.
copy themselves.
More
So now
like
we
no
actually have a
longer.
4n cell at this
Accident?
point.
No,
But since we're
not
doing
accident.
meiosis, this is where actually
Where
crossing
are
over
we?
occurs.
I'm not
And
really
so
my
parts
own.
of this chromosome
Where's
will
the
swap
real
places
kid?
with this chromosome and vice versa. What that gives
You're the
us
ones
is
in Alice. You're
variability
Emily.
in all of the
You're
sperm
what-
and the egg.
What was that?
They'll divide in half.
We're
Now we have
70
2n
bit.
cells,
You're
and then
7
eventually
a
we'll have n
bitch.
cells equals
You're 7 a bitch. Oh no, no,
2.
no, no! You
And
got
so
me
we started with
all
2n
over
equals
the
4,
place.
now we have
don't
n equals
have to
2. Each of these 4
familiar.
things in a
Familiar.
male
And
become a
that's
sperm,
longer.
and in a
We're
female,
having
one of them
longer
will become an
place.
egg. And the
Please
other
don't
ones
leave her
will
alone
actually
with
form
her.
polar bodies. And so what do we get
let's
after
see here now.
meiosis? Well, we get
Here's the
sperm,
right
and we
here
get
for
egg.
what I know.
And
It
we can
was
then
different
go
now.
from
Now-
these original
This is the
2n
number
equals
one
2
day.
to
It's the number
eventually
today.
or n
Yeah,
equals 2
what
to a 2n
more?
equals 4
You're not
zygote.
really all my
And so now we're
friend.
back to a diploid
No.
cell. And
Please,
that diploid cell
don't
can enter into the
touch
cell
me
cycle.
alone.
This is a zygote to make
Please
more cells
don't
and
touch
eventually
me here.
make sex
No,
cells to
please
make
don't
the next
leave me alone
generation.
with her.
So
No,
that's
it's
cell
not
cycle,
getting back.
mitosis,
I'll be right
meiosis,
back
and I hope
before
that's
I know
helpful.
it.